Welcome to the Cosmos Hub, a suite of interactive, cloud-based analytical tools specifically designed for microbiome researchers. The Hub can accept raw FASTQ data from a variety of sources, such as dragging and dropping them from your local machine or uploading them from a variety of other integrations. You have the ability to select multiple workflows and upload your metadata to enable comprehensive comparative analysis. There are several pipelines available on the Hub, which can be read about in our documentation. These include a variety of applications for human and non-human specific studies, as well as functional, AMR and virulence, as well as amplicon, long read and short read pipelines, in addition to a number of comparative analyses for reporting and interpreting your data. Navigating the Hub is very simple. It's organized in a file-based structure, which you can organize based on sample type or study. You can select individual samples and find the lists of taxa that were identified. You have options to switch between species levels. You see stats about the number of reads produced, as well as the number of host reads that are detected based on the pipeline that you run. There are a number of visualizations that are used to analyze individual taxonomic and functional profiles, and everything is version controlled, so you can see different versions of the pipeline as it updates. You can also view the information about functional modules. We also have information about FastQC for all of your data. And each of these tables can be exported as a PDF or a TSV, where you can combine multiple samples for comparative analysis. Within the sample viewer, you have the option to share data with your collaborators. You can also download raw files and samples, as well as multiple taxonomic profiles. For each of your samples, you have the option to add comprehensive metadata to describe these samples, such as day, age, sex, BMI. Once you have the relevant details available for your samples, you can edit the individual files individually, creating custom fields such as treatment groups. You can also select multiple samples and edit all the metadata through a template CSV. Once your metadata is integrated, you can leverage these labels to create a comparative analysis. Once you do this, you can separate your samples into individual groups based on the metadata variables and begin to ask questions such as comparing taxa across cohorts, as well as functional AMR pipelines. We also have differential abundance tools such as Moslin and Lefse, as well as multi-QC reporting. You can view your comparative analyses in as many different versions as you'd like. Or here you'll see an interactive table, which represents the matrix of all of your samples, with your individual samples abundances listed in columns and the individual taxa listed as rows. This interactive table can be sorted, can be searched. It can also be exported as a TSV, which is used for downstream reporting and other tools. You have a variety of visualizations to support your analysis, such as interactive heat maps, stacked bar charts, and even abundance distribution. We also have ecological tools such as alpha and beta diversity with a number of metrics to choose from, as well as built-in statistics to run the correct statistical tests needed for publication. Each of these figures can be exported as raw data or in image formats. You also have the option to generate these visualizations for your functional results as well, which will give you an overview of the functions that are being performed and your metagenomic sample. You can also leverage our differential abundance tools, such as Moslin, to generate association tables, which shows which taxa are enriched in which cohort and to what level, with accompanying P and Q values. You also have this data represented in heat maps, volcano plots, random forest plots, and blocks plots and scatter plots. You can also leverage our new AI tools such as Ask Rita to ask questions about your hypothesis in relation to particular taxa and functions, where it'll give you a detailed overview of your query with primary literature citations to support rapid interpretation of microbiome analysis data. Lastly, you can consult our documentation, where we have a number of videos, instructions, and details about our pipelines to how to get started. With the Cosmos Hub comes a suite of support technicians that can assist you with analyzing your data, navigating the platform, or helping you ask microbiome-based questions. Thanks for a tour of the Cosmos Hub. If you'd like to try out the software today, you can go to cosmoshub.com and click Book a Demo. Thanks for your time, and we look forward to you analyzing with us.